Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And welcome to the live sessions where you can get an opportunity to ask the questions and you can clarify your doubts. And it's very uh, good opportunity for you to interact with me, and I'll be happy to answer your questions. So if you have questions, you can just write into the chat box and we will be happy to discuss about that. Uh, so I don't know the name of this guy which says science is everywhere. Can you spell out your name? It looks very strange actually, the kind of name what you're using. But anyway, what you're asking is the how to calculate the vibrational frequency of a nucleotide base pair. So when you want to calculate the vibrational frequency, you have to use the Priyanka. Okay. Priyanka, can you elaborate your questions? Maybe you can give me an example or something. I don't know what, uh, without any information, how we can calculate the vibrational frequency. And in what context you want to know actually about that also. Priyanka, very frankly, I am not an expert of IR spectroscopy. Okay, but still, my your question is not clear to me. You said every bond has a unique vibrational frequency. That is true, but uh, how we can use that to calculate the vibrational frequency? I think better you can discuss this about someone who is more an expert of the IR spectroscopy. I am not actually. I am. I have. We have. I have. We have discussed very briefly about that particular topic, and I am not an expert of that. Priyanka, I think I have already given the link. When if you see my presentation, I have already given the link for the NPTEL course, the NPTEL web course what we have developed, and I think in that we have discussed about that how to calculate the vibrational frequency, and if required, I think I will share the additional resources with you, and just drop the mail into my discussion box, and I'll be happy to help you. Priyanka, I think we have. I think the NPTEL is trying to make a receptory, and where they are actually going to have the exam. So, in any case, the exam, if you attempt the assignment number 12, okay, those 20 questions are actually going to be very close to the kind of the question paper what you're going to get, except that in the sample, in the question paper, the real question paper, you're going to have the 100 questions. Here we are only providing you 20 questions. And this assignment 12 is actually somewhat very closer to what the sample paper is going to be look like. If you want to attempt the previous year's sample papers, then that also we can upload onto the website, okay, and then you can be able to attempt, or you can be able to see what kind of questions you are going to uh, encounter, okay. So we'll do that. Priyanka, I think we don't have any such course or any such plan, but I think if you are interested and if there are online, if there are hands-on training sessions conducted by the CET, like under the QIP program or under the QIP program, and if you are a faculty, you can be able to join those programs. Even if you are a student, you can join those programs by paying the fees also. So I think there are programs, but those are not related to NPTEL, but they are more related to the individual IITs where the TQIP or other such kind of programs are there. So if there are any such program, I think they will be posted on the website of the individual IIT's CET, right? And you can join them. Either if you are faculty, it may be free. If you are a student, you might have to pay a nominal fees also. Priyanka, which website you're talking about? Means like the CET's website or uh, so IIT Guwahati CET website is anyway given on the IIT, and you can browse the website and get the information about the different types of TEQIP program which they are going to conduct or which they are going to run. And if you find them suitable, you can join them. You can, I think you can easily join the gene editing courses because the basic information anyway we have discussed how you can be able to isolate the genes, how you can be able to clone them. So I think this basic information is good enough, and I think we have taken we have actually shown you many demo also. So by this, I think it should be good enough for you to join these gene editing course run by the companies, and it may be helpful for you to join those courses. Hello, student. If you have any questions, you can ask me. Otherwise, our live session is very limited. And if you will not ask me questions, I think I will conclude the sessions, and then you will not be able to, have this opportunity to clear your doubts, because this is very important for you, especially when we have an exam in another 2 weeks. Okay. Um Priyanka, what you want to know about the back mutations? So, what is mean by the back mutations? Back mutations is like you suppose we have a wild type gene, right? And it got mutations. So then it is actually going to have the mutated gene. If this mutated gene is actually going to have the additional mutations, right, which are actually going to reverse this process. For example, if you have AATG and if that ATG is going to be get converted into AAG, then that is called a mutations. But if and then you're going to have the mutated gene where you're going to have the AAG. But if you have the mutations and then you can develop or you can actually reverse the things, then that is going to be called as back mutations. And it actually is going to uh, oh, okay, so now what you are asking is how we can do a back mutation in lab. So I think we discussed how we have not I think discussed how you can be able to do generate the mutations, right? But generating a mutation is very easy. So what you have is, for example, if we have a DNA sequence like this, A, T, G, C, 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 A, T, 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 like that. Okay. So this is the 5 prime, this is the 3 prime, and I'm not showing the second sequence. Okay. And for example, if I want to change this C into G, okay, or suppose by mutation this C got changed into G, right? So what will happen is you're going to have the gene which is going to have this sequence. Now, if I want to change this to this, okay, I can do that in the lab. So what I have to do is I have to change the G to C, right? So what I'll do is I'll generate a primer and I'll do a long PCR method. So what I'll do is I'm going to generate a primer like this, A, T, G, C, C, and instead of G, I'll put the C and I'll put A, T, 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 okay? So this is going to be my forward primer, right? Similarly, I can have the reverse primer, which is also going to be complementary to this. And then what I can do is I can do a PCR, okay? So when I do the PCR, I am going to get the mutated gene, which is going to be mutated back, right? And so in one place, this is not going to match, right? So in one place, it is not going to have a mismatch. So in this place, it's going to have a mismatch. And then I'm going to have this mutated genome or the mutated gene. And that mutated gene I can clone into the new vector. I'm sure we have already discussed the different types of procedure, how you can be able to clone. So it's a very simple thing. If you have to do a PCR, and so this is the region where you have actually put the forward primer, right? So if you put the forward primer and you put the reverse primer on this, right, you're going to get Hello. Okay. Okay. So this is what I said. We can have this gene. We can have the if this got mutated. So this is going to be a mutated gene, and in that mutated gene, if you generate a primer, so that forward primer is going to be this one, the reverse primer is going to be this one. If you like, you can have the mutations in the reverse primer also. That way, both the copies of the genome is going to be both the copies of the gene is going to be mutated. Okay, but that is not required because you can easily clone this gene, because if it's go for the multiple round, then this itself is going to give you the two copies, right? It's going to give you the wild type, right? It's going to give you the wild type, and it's going to give you the mutated, right? This mutated one is actually the back mutation, so it's going to give you this one, right? Whereas this wild type is actually a mutated version, right? And this one is mutated, but this is going to be wild type at the end. Okay, and then you take this gene and clone it into the new vector. Okay. I hope it is clear to you, Priyanka. If it is not, I think we can discuss further about this. Um, I think if we don't have any more questions, Priyanka, do you have any more questions? Okay, I think since we don't have any more questions from the student side, I would like to conclude this session, and um I'm happy that you have taken this course, and I'm sure by attending the material what we have generated and by interaction through our various live sessions, it would have been benefited you, and I hope at the end this course would be helpful for you. So all the best for your exams, and with this I would like to conclude my live session.